Hello everyone, my name is Paul Waynes. I am the Senior Laboratory Technician up here at the Derriford Research Facility. So I help in the management of these biomedical research labs, both in this building and in the John Ball building. One of the pieces of kit I'm responsible for day to day is this lovely piece of kit here, which is a Fax Aria 2. Now the fax element of it stands for fluorescence activated cell sorter, and that kind of gives you an idea of its capabilities. So I'm just going to talk a little bit about that before handing you over to my colleague Marwa, who I think you may have seen before. Marwa is going to talk you through her particular research with a focus on cell cycle analysis, which is one of the many applications that this machine is useful for. I'll try and give you a flavour for what it can do as we go on. Okay, so as is the case with all flow cytometers, we've really got three elements to it. We have the fluidics portion of it, which comprises the fluidics cart here and also the components in here, which I'm going to show you in a minute. We also have the laser side of things, which obviously is to bring about the fluorescence that the flow cytometer picks up and turns into data. That is all kind of tucked away in the bowels of the machine here, but there's a small part of that I can show you shortly. And then finally we need to visualize all this data that we're gathering, and we do that on the software here. So there's really, those are the three kind of core elements of flow cytometer. As I say, this one has a bit more capability than your normal flow cytometers. I'll talk to you a little bit about that. But first off, let's have a look at the fluidic side of things. So what we have here, going from left to right, is we have a waste container here, because obviously as our cells and our sheath fluid, our ethanol, etc. are going through the system, a lot of it is going to go to waste. That's what we have in here. This small container here contains 70% industrial methylated spirits, or 70% ethanol, whatever I have to hand. This is used for sterilizing the machine, which is a very important process to do once you've finished with your analysis or your sorting, whatever you were doing. And then in terms of actually running your cells or your sample through the machine, probably the most important component here is our sheath fluid. Sheath fluid, really in simple terms, is basically PBS with some stabilizers in it. And the purpose of the sheath fluid is essentially to carry your sample through the flow cytometer. I'll talk a little bit more about that shortly. So this is your fluidics cart. The reason it's making so much noise, and I hope you can hear me above it, is because there's a compressor built into it, which is actually producing the compressed air, which pushes the liquid around the system. So instead of your sample being pulled around the system, being pumped around it, it's actually being pushed around the system by compressed air, which ultimately gives you a lot more fine control over how you run your samples through the flow cytometer. Carrying on the theme of fluidics, we've now opened up the flow cytometer to show you what's inside. If I just point out a few components, and I'll try to explain what happens inside here to enable you to gather your flow cytometry data. So I guess the most important part of this is the flow cell here. Now the purpose of the flow cell is to enable us to get our cells into a nice straight line so that they can be interrogated individually by the lasers that we have built into the flow cytometer. I'll talk more about the lasers in a minute, but suffice to say that this is the sort of the crucial point of the flow cytometer in effect, because this is where your analysis happens. How does it happen, I hear you ask? So what actually is happening here is we have sheath fluid coming from the tank down here. It's coming in at this point and this point here. And at the same time, we have our cells coming in from a sample chamber here, traveling through this tube here and into the top of the flow cell. Now, the purpose of the sheath fluid is to focus those cells from what is essentially a jumble of cells when they first enter the flow cell into a nice straight line, so that the flow cytometer can interrogate each one in turn. How it does that is by a process called hydrodynamic focusing, and essentially that occurs as the cells travel down through this, I don't know whether you can see it, but there's kind of an inverted cone there, and the shape of that is such that it facilitates this hydrodynamic focusing. Once those cells are in a nice straight line, they pass down through a cuvette here, where they're interrogated by, in this case, both a blue and a red laser. And the sort of the final steps of their journey, and they pass through this, what's called a nozzle here, and then finally they run out through the nozzle and they run to waste here. I've got an example of a nozzle here. This is exactly the same as the one that's in place there. This is essentially a piece of ceramic, and you can see a tiny red O-ring there, which is around a hole which passes through the nozzle. Not all flow cytometers work in this way, but because this one has the capability of sorting cells, it's kind of a crucial element of this particular machine. So as your cells pass through the nozzle, having been analysed, each cell ends up in an individual drop, which is, you can see here, falling down to waste. It looks like an it looks like a kind of a solid stream of liquid, but actually it's not. It's a stream of drops, which if we go over to the screen, we can actually see those drops here as a binary image. So in terms of the way that these cells are actually analysed or interrogated by the lasers, I'll try and explain a little bit about that. As I mentioned, this machine is equipped with a blue laser, which has a wavelength of 488 nanometers, and a red laser, which has a longer wavelength of 633 nanometers. You can't actually see the lasers as they're generated, obviously for safety reasons. That is actually done in the bowels of the machine here. You'll find other flow cytometers have other laser wavelengths available. You can have UV lasers, violet, virtually sort of covering the whole visible colour spectrum. But one thing I want to show you on the laser front is this part here. So I've mentioned previously about cells being interrogated in turn by the lasers. how that actually happens is the lasers will fire perpendicular to the stream that the cells are in, and as the cells pass through the path of the laser, we get a phenomenon called light scatter. Two types of light scatter: forward scatter, which is scattered light in a forward direction, and side scatter, which is scattered light which is perpendicular to the path of the stream. And they are telling you two slightly different things. So in the most basic terms, the flow cytometer can tell you things about both the size and the internal complexity of the cells that you're looking at. So forward scatter gives you an indicator of the size of these cells relative to one another. Side scatter gives you an idea of the internal complexity of those cells. So that is independent of size. We can look at that when we look at Marwa's samples. We can show you that in a bit more detail. And then taking the analysis side of things a step further, if you've treated your cells appropriately, be it with a fluorescently tagged antibody or with a fluorescent molecule that, for example, binds with DNA, then we can use those lasers and the various other components of the flow cytometer to measure fluorescence. And we use that fluorescence to tell us various things about the cells that we're running through the machine. So in terms of experimental design, your strategy is very much based around how you use that fluorescence to tell you what you need to know. So I mentioned about forward scatter, I mentioned about side scatter, I mentioned about fluorescence. It's important that all of these signals are gathered and translated into data. Now, the way that happens is, well, obviously it's an extremely complex process, but essentially, as I mentioned, we've got laser light going across perpendicular to our stream. Any scattered light, any fluorescence is then picked up and kind of channeled by the flow cytometer via fibre optics into what are called laser arrays. So we've got an array that serves our blue laser, and we've got an array that serves our red laser. I've got a bit of a question for you. I mentioned that we have a blue and a red laser on this machine, and I mentioned the blue laser has a fixed wavelength, 488 nanometers, and I mentioned that the red laser has a fixed wavelength slightly further down the visible light spectrum at 633 nanometers. How do we actually utilize just those two lasers to gather multiple bits of information from a single cell? Just have a think about that and see what you can come up with. What is actually happening in here, all of that transmitted light, so all of the fluorescence, all of the scattered light that occurs as a consequence of the laser firing across the cells, is bounced around in here amongst an array of filters and mirrors, and depending on the wavelength of that light that is emitted, the wavelength of the light that is emitted will dictate whereabouts in this array those signals are picked up. So we have, for example, in the blue array, we have 6 possible locations where slightly different wavelengths of light can be picked up and translated into electrical signals via these photomultiplier tubes. In the case of the red laser, we have two possible wavelengths that it can pick up. So we've got a total of really at least 8 different colours, for want of a better word, that we can actually visualise at any one time. Okay. I introduced this machine at the beginning as a fax aria 2. The fax part of that, if you remember, stands for fluorescence activated cell sorter. Now, the cell sorting side of things is really the main reason why this machine is as big as it is, and I'll try and just give you a very brief explanation as to how that works. Essentially, what the machine is able to do, it's able to analyse a single cell, and it can analyse up to probably 10,000 of these cells every second, and with each individual cell it's capable of deciding, according to whatever parameters you kind of you give it, it's capable of deciding whether or not it wants to keep that cell or sort that cell into a tube or onto a plate or into a 96 well plate. There's a whole variety of containers you can actually sort individual cells into. So where this might be useful is if you have a mixture of cells, some of which are expressing GFP, most of which aren't expressing GFP. What you could potentially do is you could tell the machine, okay, I only want to keep the GFP expressing cells. So those GFP expressing cells would be fluorescing as a green colour, which the machine would pick up when it analyses the cells. So with cell sorting, what it actually does is able to analyse each of these cells in turn, see whether it's green fluorescing or whether it's not. If it is green fluorescing, it's going to know it needs to sort it, and it can actually make calculations and it can make adjustments to make sure that when that cell enters a particular drop in this stream, it can actually change the characteristics of that drop to sort it into a container of your choice, and then you can take that container away, be it a tube, plate, whatever, and you can culture that cell. You can culture that cell on in the lab. So in the context of let's stick with the GFP example, what you can then do ultimately is increase the number of GFP expressing cells that you have by culturing them, maybe doing another sort to purify out the non-GFP expressing cells, and over time what you would hope to end up with is a pure culture of GFP expressing cells, which in turn have other characteristics that you might be interested in. So the way it does it is carries out its analysis, and then it very cleverly calculates the time it takes from when the cell is analysed to when the cell enters this stream of drops. So effectively what the machine does is it focuses all of its attention on that drop there. So by virtue of the calculations that it's made after the analysis, it can work out when your cell of interest is going to appear in that drop that I just pointed out. When it sees it, it will apply a very small electrical charge to that drop, to the surface of the drop, so the cell itself inside is unaffected. but by applying a small electrical charge to that drop, it can actually facilitate the movement of that drop out of this main stream here. At the same time as doing the, while we're doing the analysis, what we'll also have done during the setup of the machine for sorting is we will have turned on these plates here. So we've got plates here, and what these plates are doing is they're effectively framing that stream, and they're both charged with about 2,500 volts going through. So what happens is the interaction between the charged drop and those two charged plates brings about the movement of that particular drop with that cell in it out of the mainstream. So it kind of swerves out of the mainstream. All the other drops go to waste, but that drop will fall into whatever container you've put in there. So you can put in one of these fax tubes here. You could sort into a Falcon tube like this. So there's a lot of flexibility with regards to how many cells you collect. You might only want to collect one cell, you might want to collect thousands, but you have an awful lot of capability with what exactly you can collect. So you might want to collect 4 different cell types from one sample. You can do that. And just to demonstrate to you how it creates these side streams that it uses to sort these cells of interest, if we put these plates on, what we can actually see here is we can see we have a red laser firing across the stream, and hopefully you can see 4 side streams which have been created because of that interaction between the charged drops and the charged plates there. Now I'm not going to and those side streams you can see on the screen at the bottom there as essentially white dots in that case. And that essentially what you would do is you would then set up the system to optimize your sorting and make sure that you are maximizing the recovery of your cells. I'm not going to leave that open for very much longer, because obviously we have live plates, so for safety reasons I'm going to close that. So that's just a very brief overview of how the cell sorting is done. Obviously the setup etc. is a very complicated process, but it's an extremely useful tool in an extremely wide range of circumstances. So I've talked you through the fluidics side of the flow cytometer. I've also talked you through how the lasers work in the context of analysis, but all of that is useless unless we have a way of actually looking at the data that we're gathering, and obviously the software is how we do that. So I'm going to give you a very brief overview of how the software works, and I thought it would probably be easiest to do that if we do it in the context of actually loading and looking at a sample in kind of very basic terms. So what we have here is a cell sample that Marwa has produced. She's treated it with propidium iodide, so we should be able to see it on the flow cytometer on the software. I'm just going to show you how we would load a sample, and then briefly talk you through the different elements of the software. So if these cells have been sat in a tube for a while, there's a good chance they're going to have settled down to the bottom of the tube. Ideally we want them in a bit of a suspension, and the best way to do that is with a good old vortex mixer. I say old because that vortex mixer looks like it's older than me, but it still works. Okay, so we're loading it into the sample chamber, and then if we focus on the software, you can see that on this left hand screen we have what's called an acquisition dashboard. And what the acquisition dashboard enables us to do is to load our sample, acquire the data, record any data that we might gather. So if I just click on load, we want to be focusing on the tube here. Okay, so the tube now has disappeared up into this chamber here, and if you remember back to when I was talking about the fluidic side of things, the sample is being pushed through and into the flow cell, where it's now going to be analysed. So in terms of the different elements that we have here, if we focus on the left hand screen first, we have our we can see our stream. Now our stream is kind of far more crucial to us if we're doing cell sorting, because we have to make sure that that is as stable as possible. But if we're just doing a straightforward analysis, as we are here, it's not so important, so I'm not going to talk too much more about that. What we have in the middle, as I said before, is our acquisition dashboard, so we can load and unload samples, we can start and stop the acquisition of our data. We can, when we're ready, we can record data, and we can record any number of events that we wish to record. And it's also talking, and it's also showing us how many events we have going through the flow cytometer and being analysed every second. Now you can see that currently we have around about 250 average events per second, and I just want to talk very briefly about that term events. Up until now I've been talking about cells going through the flow cytometer, but it's actually showing us events, and it's very important to distinguish between that term events and the term cells, because not everything you see in on this worksheet here is going to be cells. Some of it is going to be parts of cells, it's going to be big lumps of bits of cells, cells that haven't quite finished dividing, whereas really in most cases what people will be most interested in is nice single cells. But the flow cytometer relies on you to be able to translate what you're seeing so that your focus is on single cells. It will record everything as an event. And so as a person doing flow cytometry, it's important for you to be able to recognize what you need, what you don't need, and focus on your single cell population. Other elements of the software, we've got our files with our folders and experiments in. So we've got one open at the moment that Marwa's going to talk to you in more detail about in a minute. But really, in terms of data gathering, we want to be focusing on this screen here and working kind of from the top left. What we have here is called a scatter plot, and on the scatter plot we can see our events, so our cells and our debris, everything else, represented as little dots here, scattered over the screen. And you can see that the cytometer is measuring FSC, or forward scatter, which I talked about earlier, and SSC, or side scatter, on the y-axis. Now the forward scatter, if you remember, is an indication of how big these cells are, how big these events are in relation to each other. So as we move up the scale here, our cells, our events are getting larger. And side scatter, in contrast, is looking at the internal complexity or the granularity of our events or our cells. So as we go up this scale here, our cells or our events are becoming more internally complex or more granular. So we can make very broad kind of generalizations about the morphology of our cells using this scatter plot here.
Hello everyone, my name is Marwa Jbara. I am a PhD student. My research focuses on the impact of some plant species on mantle cell lymphoma, and in our experiment today we are trying to study the impact of some of these essential oils on REC1 R288 cell line, which represents the primary stage of mantle cell lymphoma. So to do the cell cycle analysis, we are using the flow cytometry. And as Paul mentioned before, how to load our samples and recording our data, we set up the first plot here, forward scatter versus side scatter, and the reason of choosing the forward scatter because it gives us information about the cell size, while the side scatter gives us information about the cell's internal complexity. So here we gated the main body of our cells, trying to avoid the debris, which has a smaller size. And from this first population, we generated the second population using the width versus area, trying to avoid the doublets, because doublets generate bigger signals than single cells. And from the second population that we suggested that it's just the only cells, we generated the histogram. And in the histogram here, we have B3, which stands for the G0, G1 phase of the cell cycle. And then the B4, which is the synthesis phase, and B5, which is the G2M phase. And how we can set our markers and differentiate between these phases. As we know, in the cell cycle analysis, cell cycle at different stages of the cell nuclei have different amount of DNA. In our experiment, we prepared the samples and treated them with the propidium iodide which binds with DNA. So in the G1 phase, cells have 2N, while in the synthesis phase, cells have more DNA than G0, G1 phase. And in this G2M phase, cells have twice the amount of the DNA that have in the G0, G1 phase. This is why this x-axis stands for the propidium iodide fluorescence versus the count of cells. And in this statistics table here, it gives us the percentage of the cells in each phase. So for example, our population that we are interested in, which is the single cells, is B2, the second population, while the B3 is the G0, G1 phase. So here we can see that 61% of the cells are in the G0, G1 phase. B4, which is the synthesis phase, 20% of the cells are in the S phase, while 12% of the cells are in the G2M phase. So here in this sample, I will show you the impact of some of the components that we are working on at different concentration on the cell cycle. So compared to the control sample, in the histogram we can see here that there's a sub-G0 G1 phase. So the component that the cells are treated with affecting the cell cycle of the cells by causing the sub-G0 G1 phase. And in this table here, it gives that 90% of the cells are in the sub-G0 G1. And this means that this essential oil causing apoptosis, so the apoptotic cells in sub-G0 G1 phase